Welcome to this Pearl of Laboratory Medicine, brought to you by AACC and the Clinical Chemistry Trainee Council. View this and many more pearls, as well as other free educational material at traineecouncil.org.
Hello, my name is Olena Morozova. I'm a clinical molecular genetics fellow at the University of California, San Francisco. Welcome to this Pearl of Laboratory Medicine on classification and genetics of sarcomas. Sarcomas are a heterogeneous group of tumors of mesenchymal origin. Sarcomas can be grouped into two main classes, bone sarcomas and soft tissue sarcomas. There are more than 100 different histopathological subtypes of sarcomas defined, and about a half of them are associated with characteristic somatic molecular abnormalities. Often, those abnormalities are gene fusions. In addition, sarcomas are often associated with germline predisposition syndrome. Bone sarcomas are very rare and represent less than 0.2% of human cancers. There are about 3,000 diagnoses and 1,500 deaths a year. Bone sarcomas often occur in older children as well as adolescents and young adults. Most common subtypes of bone sarcomas are osteosarcoma, and that's about 35% of cases, chondrosarcoma, that's about 30% of cases, and Ewing sarcoma, that's about 16% of cases. Ewing sarcomas are associated with a gene fusion involving EWS and SLY1 genes. Soft tissue sarcomas are a bit more common than bone sarcomas and represent less than 1% of human cancers. There are about 12,000 diagnoses and 4,500 deaths a year. Soft tissue sarcomas are diverse, with over 100 different subtypes that can occur in different age groups. Roughly half of the soft tissue sarcomas are associated with a recurrent cytogenetic abnormality. Some subtypes with associated genetic abnormality are listed on this slide. Now, the next few slides show examples of different types of sarcomas. So, for example, here is an H&E image demonstrating a monogamous population of small blue round cells in a soft tissue tumor. This is an example of a Ewing sarcoma. The next H&E depicts a well-differentiated liposarcoma with variable-sized adipocytes with nuclear atypia. This slide shows an H&E demonstrating a proliferation of atypical spindle cells with laying down of malignant osteoids. This is an osteosarcoma. And finally, this H&E shows an atypical fascicular proliferation of spindle cells with increased mitotic index. This image is characteristic of an undifferentiated pleomorphic sarcoma, or also known as UPS. Sarcoma genomes fall into one of three classes: those with a simple karyotype with a defining translocation or gene fusion, those with a simple or complex karyotype with a specific mutation, and those with a complex karyotype with multiple chromosomal rearrangements, duplications, and deletions. Gene fusions are common molecular events in sarcomas. As a result of some sort of genomic rearrangement, two genes are juxtaposed together. They're then transcribed as one chimeric product, creating a fusion transcript. Gene fusions can result in a loss of function of one or both partners, a gain of function of one partner, or a completely novel function. Gene fusions can be detected using a variety of molecular assays. Fluorescence in situ hybridization, or FISH, specifically break-apart FISH, is a molecular test used clinically for detecting gene fusions in sarcomas. This test employs two fluorescent probes of different colors that hybridize to two different regions on a chromosome. The two probes are designed such that they map proximal and distal to the fusion breakpoints in the gene of interest. When the probes are fused, it indicates a normal gene. If the probes are apart, it indicates a break in the gene irrespective of the fusion partner. Specific gene fusions can also be detected using PCR, performed on either the DNA or RNA material. Next-generation DNA sequencing can be used to detect a variety of different fusions in a sample. Gene panel sequencing approaches can detect fusions in genes of interest. An important gene panel design consideration is to include intronic sequence, because fusion breakpoints often occur in introns. Whole genome sequencing is an attractive, comprehensive approach which is currently used in the research setting. And finally, RNA sequencing can be also used in a panel format or as a whole transcriptome to detect both known and novel fusions. RNA sequencing may have an added advantage of being able to quantify the amount of the fusion transcript. This image demonstrates sarcoma positive for an EWSR1 rearrangement assayed by break-apart FISH. Green and red fluorescently labeled probes flank the EWSR1 locus. Cells negative for an EWSR1 gene fusion have two yellow signals produced from the overlap of the green and red signals. Cells with split red and green signals are positive for an EWSR1 gene fusion. This slide illustrates the principle of detecting gene fusions using next-generation DNA or RNA sequencing. In both of these approaches, sequencing reads are mapped to a reference genome, and then the alignments are examined to assign the reads into three different categories. An important note here is that paired-end sequencing, where nucleic acid fragments are sequenced from both ends, is typically used for detecting gene fusions. Concordant reads are those that have mate pairs or reads from the two ends of the same fragment mapping to the same gene. Split reads are those that map across a fusion breakpoint. These reads may be lost when mapping to a reference. And spanning reads are those that have mate pairs mapping to two different genes, and the genes would be fusion partners. The detection of both spanning and split reads would indicate the presence of a gene fusion. Molecular testing has several applications for the management of patients with sarcomas. For example, arriving at diagnosis for cases with ambiguous histology, and for example, diagnosis of small round cell tumors that have similar appearance under the microscope, refining molecular subtype of the disease, for example, alveolar subtype of rhabdomyosarcoma, which is associated with Pax-Foxol fusion, while embryonal rhabdomyosarcoma is not. Identifying treatment options, for example, KIT and PDGFRA mutations in gastrointestinal stromal tumors, and those are associated with response to imatinib. Confirmation of lesions with an unusual presentation, for example, confirming a synovial sarcoma in a 76-year-old individual. And of course, synovial sarcomas are usually pediatric neoplasms. And finally, aiding in diagnosis for cases with dedifferentiated histology. I would like to highlight further one particular application of molecular testing for the management of sarcomas. Small round cell tumors have homogeneous light microscopic appearance, and they look like small round cell neoplasms like we saw on one of the previous HME slides. Several entities may present in this way: Ewing sarcoma, rhabdomyosarcoma, mesenchymal chondrosarcoma, desmoplastic small round cell tumor, round cell liposarcoma, poorly differentiated synovial sarcoma, and finally neuroblastoma. Molecular testing can be used to identify a characteristic gene fusion that can help make the diagnosis. For example, the identification of an EWS-Y1 fusion would be diagnostic of a Ewing sarcoma. Several cancer predisposition syndromes are associated with an increased risk of sarcomas. Li-Fraumeni syndrome, caused by germline heterozygous inactivating mutations in TP53, is associated with childhood onset sarcomas. Retinoblastoma syndrome, caused by germline heterozygous inactivating mutations in RB1, is associated with osteosarcomas. Familial adenomatous polyposis, or FAP, caused by germline heterozygous inactivating mutations in APC, is associated with desmoid tumors. There are also several genetic syndromes which have many different phenotypic manifestations, including a predisposition to sarcomas. Some examples of these conditions are listed on this slide. So here are some take-home messages from my talk. Sarcomas are a heterogeneous group of diseases with over 100 subtypes defined by histopathology. Genetic testing can help with the diagnosis, molecular subtyping, and treatment selection. A significant fraction of sarcomas are associated with either a germline cancer predisposition syndrome or a genetic syndrome with other phenotypic features. Thank you for joining me on this Pearl of Laboratory Medicine on classification and genetics of sarcomas.
For more like this, as well as articles, podcasts, and more, please visit the Trainee Council at traineecouncil.org.